All right, now that we've discussed the shape and structure of the organelles and a couple of the processes like cellular respiration and photosynthesis, now we're going to move on to the cell cycle and cellular reproduction. In this chapter, we're going to cover the cell cycle, the eukaryotic chromosome, mitosis and cytokinesis, the cell cycle in cancer, and then finally prokaryotic cellular division. The process of cellular division is a highly regulated one. Life begins with single cells, but in only a very short amount of time, that cell can lead to trillions and trillions of cells through the process of cellular replication and division. Not only can these single cells produce millions and trillions of cells, they also give rise to highly specified cells. So even though you start out as a single-celled organism, there are over 200 different types of specialized and individualized cells in your body, each with their own unique function to perform. This process of cellular replication and division is what we're going to be studying next. So we're going to kick things off by studying the cell cycle. The cell cycle is an orderly set of stages from the first division of a eukaryotic cell to the time the resulting daughter cell divides. So just before the cell cycle begins, it has to undergo a series of transitions. First, it grows larger. The number of organelles will double. The cells will have to undergo DNA replication, which leads to a doubling of the genetic component. This first stage of growing and doubling is the first of two major stages of the cell cycle and is what's known as interphase. The second stage is called the mitotic stage, which includes two phases, mitosis and cytokinesis. Oh, so here you go, notes for all of that. So like I said, the cell cycle is going to be a very orderly set of stages for that. That starts with the very first parent cell division and ends with the daughter cell division. Before that division, the cell grows larger, the number of organelles doubles, DNA is going to be replicated, and then here are the two major phases of the cell cycle. This diagram here is excellent. It goes through not only the two phases of the cell cycle, but breaks them down into their smaller components. So here we have interphase. Interphase, like I said, is the stage before we begin cellular division. So interphase is where we are going to see growth. The cell is going to double in size. The DNA is going to double. And then we are going to prepare for cellular division. The next step is going to be mitosis. Mitosis, or the mitotic cycle, the mitotic stage, is going to involve both the division of the genetic component, which is mitosis, and then division of the cytoplasmic component, which would be cytokinesis. So the first thing we're going to look at is going to be the stage of the cell cycle known as interphase. Most of the cell's life cycle occurs during this phase. In fact, it can take up about 90% of the cell cycle. So this is where and when the cell is going to perform all of its usual functions depending on the cell type. So if it's a liver cell, it's going to be busy with detox. If it's a kidney cell, it's going to be cleaning out your bloodstream. If it's a pancreatic cell, it's going to be producing insulin. All of that happens during interphase. So we're going to perform our usual functions and we are going to get ready for mitosis. Mitosis is going to be an orderly set of stages where we're going to divide the genetic component as well as the cellular component. But interphase, the beginning part, has three stages or three phases. The G1 phase, the S phase, and the G2 phase. We'll go through each one of these. So here are the three different phases, G1, S, and G2. The G1 phase is going to be that first initial phase. This is also known as the growth phase. During this phase, the cell is going to recover from the cellular division that just occurred. The cell is going to increase in size. It's also going to increase in the number of organelles like mitochondria and ribosomes, and it's going to accumulate the raw materials that will be used for DNA synthesis. During the rest of the time, the cells are carrying out their normal daily functions like communicating with other cells, secreting substances, things of that nature. The next phase is going to be the S phase. This S phase is where DNA synthesis or DNA replication takes place. We are also going to be synthesizing the proteins that we need for DNA replication. At the very beginning of the S phase, chromosomes only contain a single chromatid, meaning that they only have one copy of the DNA helix. But after DNA replication, there will now be 2 identical pairs of chromatids. These are going to be referred to as sister chromatids. Although they are identical, they remain attached and are going to be separated at a later stage of mitosis. So here we have an initial chromatid. During the S phase, it's going to duplicate, and then we will have sister chromatids. They're going to be attached right here at the center. We will separate them at a later stage. Then we move into the G2 phase. In the G2 phase, this is going to be the stage that occurs, or the stage between DNA replication and the beginning of mitosis. During this stage, the cell is going to make all of its proteins that will be used in cellular division. It makes the proteins that are going to be necessary for creating those spindle fibers that are going to be responsible for organizing and arranging the rest of mitosis. Then we move on to the mitotic stage. Now, the mitotic stage can be broken down into mitosis, which is the division of the nuclear material, and cytokinesis, which will be division of the cellular material. Mitosis itself can be broken down even further into 5 individual stages: prophase, prometaphase, metaphase, anaphase, and telophase. So we're going to, before we really get into that, we're going to talk just a little bit about how we regulate the cell cycle. The cell cycle doesn't just run automatically all of the time. There are a lot of checkpoints and signals that control the growth and development of the cell through this cycle. This is done through the use of signaling molecules. Signaling molecules can be either internal or external signals, meaning that they originate from within the cell or from without. So just for example, external signals are signals that do not come from the cells themselves, but are instead received by the receptor proteins that are embedded in that cellular membrane. Remember when we talked about all the different proteins that might be embedded in the cellular membrane? We talked about channel proteins and carrier proteins and enzymes, and we also talked about signaling proteins or receptor proteins that are responsible for receiving messages from other cells. These, one of the messages it might receive is an external signal that indicates, hey, it's time to undergo cellular replication. Growth factors are a great example of an external signaling molecule. There are also internal signaling molecules that regulate the movement of the cell throughout the cell cycle. And we're going to take just a quick look at a few of those. The internal cycling mechanisms, or the internal signaling mechanisms, are located at, or going to play an important role in the G1 phase, the G2 phase, and then there's going to be an additional signaling control that happens between metaphase and anaphase. Those internal signals are controlled by a molecule called cyclin. Cyclins are going to be responsible for increasing and decreasing as the cell cycle continues, and they are going to indicate we need to stop and wait until everything catches up, or no, we're good to go, we're going to move on to the next stage. Specific cyclins must be present for the cell to proceed from one stage to the next. And this is because we want to make sure before we go on to replicate the cell, we have to check and make sure that when we duplicated our DNA, we have to make sure that we duplicated it correctly. There are no mutations. There's nothing wrong. We're not missing a chromosome or anything like that. We want to make sure that the DNA is great before we go on to duplicate the cell. We want to make sure that we have lined up all of our chromosomes correctly before we try to divide them. So there's a lot of checkpoints that sort of happen throughout this process. So without cyclins, the cell cycle is going to stop at G1M or the G2 checkpoints until those cyclins can be produced, meaning that the cell is ready to move on to the next stage. Damage by solar radiation or X-rays can be repaired at the G2 checkpoint. So this is why we have these checkpoints. Any damage that might have occurred can either be repaired or the cell can be shuffled off into a process known as apoptosis. So here are the various checkpoints. The G1 checkpoint over here on the right, this is going to be the main checkpoint. If at this point the DNA is damaged, apoptosis will occur. We're not going to bother to try and fix anything. If we have an issue this early on at this stage, we're not even going to try and replicate the DNA. We're just going to shut the cell down. Otherwise, after this point, the cell is committed to divide when the growth signals are present and the nutrients are available. The G2 checkpoint over here is going to be the mitosis checkpoint. Mitosis will occur if DNA has replicated properly. If DNA has not replicated properly, we will either try to repair the damage or slough the cell off into a stage of apoptosis, which we will be talking about on the next slide. Finally, the M checkpoint is a spindle assembly checkpoint. Mitosis just will not continue if the chromosomes are not properly aligned. And we will talk about when and why and how that happens when we just start discussing mitosis. So apoptosis. Apoptosis is what is known as, or what we like to refer to as, programmed cell death. Apoptosis is a perfectly natural and essential step in the growth and development of organisms. When I was younger, they used to call this cell suicide, and I really hate that terminology. Apoptosis is not cellular suicide. Suicide is an indication that something is clearly and horribly, terribly, terribly wrong. That is not the case. Apoptosis is important, it's essential, it's natural, it occurs in healthy cells. It's just a very important part of life, and so I just personally prefer the terminology programmed cell death. It's an absolutely essential part of the growing and development process. For example, tadpoles. Tadpoles have to lose their tails to become a frog. In humans, fingers are initially webbed. Inside the womb, your fingers are webbed. Then the tissue must break down for fingers and digits to be formed. All of that is done through apoptosis. So cells that are infected with viruses need to be destroyed. Old damaged cells could become cancerous if they're allowed to grow unchecked. So this programmed cell death, this apoptosis, is a very important and healthy and natural part of the cell life cycle. Apoptosis, more specifically, what is it? It is a sequence of cellular events that bring about the destruction of the cell. This is going to be done in a very organized and very just a very organized manner. So it starts off with a fragmenting of the nucleus. So the very first thing that's going to happen is the cell is going to gather itself up in a really tight ball. This is going to pull it away from all of its neighbors on all sides. This is going to help sort of mitigate any potential damage. So it's going to gather itself up into a tight ball, pulling away from its neighbors on all sides. Then the nucleus is going to fragment and the plasma membrane blisters and bursts, and it leaves only cell fragments behind. Those little cell fragments can then be engulfed by white blood cells and other cells. And the enzymes that bring about this process are known as caspases. Mitosis and apoptosis are opposing forces. This just means that mitosis, or cellular division, is going to increase the total cell number. Apoptosis decreases that overall cellular number. Here is a diagram showing what that apoptosis looks like. Here we have a normal cross-section of tissues. And what happens is that we have lots and lots of contact between all of our neighboring cells. So the very first thing that cell is going to do is round up, become nice and smooth and round, and pull away from its neighbors. The chromatin is going to condense, the nucleus is going to fragment, and then we're going to form these little blisters called blebs that are going to sort of fragment and blister off to the side. The cell is going to fragment and contain, the cell fragments contain DNA fragments, which can then be digested by our white blood cells. So I just want to point out it's a very, very healthy, normal process. If you notice, all of these fragments are perfectly contained within the plasma membrane, so it makes them very safe and very easy to digest by the white blood cells. So that is sort of our overview of the cell cycle. We will be covering each of those stages at a little greater depth throughout the remainder of this chapter and into our next one. But please let me know if you have any questions, comments, or concerns.